Welcome back friends to the JK Biology, and today I will be described about the eukaryotic transcription by RNA polymerase sequence. Before this lecture I have described that eukaryotic transcription takes place by the three polymerases, that is RNA polymerase first, second and third. So, RNA polymerase first transcribed rRNA and RNA polymerase second transcribed mRNA, snRNA and line, and RNA polymerase third transcribed tRNA, U6 RNA, SINE and many another RNAs. So today I will be focus on RNA polymerase sequence, that is the most important for the protein formations, for the central dogma. RNA polymerase second takes place this process. Okay. So how this process would be taken, that is RNA polymerase second isolated, and it was taken in the in vitro conditions, but found that they have no any activity in vitro. So our researchers, our scientists found that another factor, that is the acting transcription factors involved and help to improve the synthesis of RNA polymerase sequence, and also it's activate the transcriptions, that is the transcription activating factor that is found distantly from the another chromosomes, that is distance from the promoter regions, and so act as trans-acting factor. Why is it called cis-acting factors and trans-acting factors? Because cis-acting factors, if the sequences is found within the DNA sequences, that is DNA here, and there is the promoter, if from the long distance within this DNA sequences act as regulatory or activatory factors, then this is called the cis-acting. But from another regions, if any factors comes, that is transcription associated factors or any transcription factors that is involved come from another regions, that is transcription factors called transregulatory. Okay. Then RNA polymerase sequence in vivo show the mRNA synthesis, because many transregulatory elements help to synthesize mRNA and improve the RNA second activity. Okay. So what is the transcription factors and what is the transregulatory element? That is the transregulatory element is found as like cyclic AMP response element binding proteins. Okay, cyclic AMP response element binding proteins binds to the cyclic AMP response element binding protein sequences, and another that is called CBF, CART binding factors. You know before this lectures that CART is the sequences act as cis regulatory element. It can also some transcription factors bind to here and can activate or repress the transcriptions or activity of the RNA polymerase second. So that is CART binding factors binds to the CART box, which is found in the RNA polymerase second promoters, that is CART box. Okay, and another here is the GC box. This is the CART box. This is the GC box. These regulatory elements found 1000 base pair or 200 base pairs longs from the promoter sites are the initiator sequences. That is, suppose here the initiator sequence denote the +1. Okay, and here found the INR sequence I have described, that is the initiator sequences found around the +1 start sites from upstream. Okay, and here the core promoter regions where the Tata box is found in the eukaryotes. Okay, that is the CART box, GC box and Tata box is the part of eukaryotic. Okay, so cyclic CART binding factors bind to the CART box. Okay, and another factors that is called CEBP. This is CART enhancer binding proteins. Okay, CART enhancer binding protein also binds to the CART box. These are the regulatory, and another is the SP1. It's activate the transcription. This is the activator of the transcription, that is SV40 SV40 early and late promoter binding sequences. Promoter binding element, okay, promoter binding element. This is called SP1. It activates the transcriptions from the 1000 base pairs or 8 base pairs, 800 base pairs longs. That is also called enhancers. Okay, so how this process would be takes place, further you will see that's which factors is involved. Transacting factors is involved for the recruitment of RNA polymerase seconds. That is transcription factor D, most important. Transcription factor D, it is also called transcription binding protein. Transcription factor second D is also called transcription binding protein. That binds to the initiator sequences and Tata box. Okay, that binds to the initiator sequences and Tata box, and it is multi subunits. That is nearly about 12 subunits. That is called transcription associated factors, TAF. Okay, that is called TAF. Transcription factor second D binds to the initiator sequences and Tata box. After binding transcription factor second D, it provide pathway to binding the transcription factor second B. It's provide the pathway to binding transcription factor second B. First points out that transcription factor second D binds to the initiator and Tata boxes, and also transcription associated factors bind to this. That's recognize the core promoters. That is the called core promoters, recognize the core promoters and also act as regulatory for the transcription factor second D. Transcription factor second F also act as regulatory for transcription factor second D and also recognize the core promoters. Transcription factor second D provide pathway for the transcription factor second B. So the reactions, that is first come transcription factor second D and transcription factor associated factors. Okay, and then again comes transcription factor second A. What is the functions? Transcription factor second A stabilizes the TAF and TF second D, stabilizes in their specific orientations. Okay, here binds and stabilizes in their specific orientation, and then transcription factor second B again come. Okay, that's act at intermediate. Okay, that's act at intermediate, and again after then what is the intermediates? To recruitments of the RNA polymerase second and transcription factor F. Transcription factor second F and followed by RNA polymerase second. Okay, transcription factor second F help to recruitment of that RNA polymerase second in the promoter regions. Okay, and transcription factor second B act as intermediate to recruitments. Okay, again then another factors, that is transcription factor second E and then transcription factor second H. Okay, E also act as intermediate for localization of the transcription factor H. Okay, localization of the transcription factor H. Then what is the functions of the H? H act as helicase. How act as helicase? Because H is the factors that's in the RNA polymerase second large subunit. There is the RNA polymerase, suppose that's multi subunits, then the large subunit contain the carboxy terminal domain. I have described carboxy terminal domain where phosphorylations occur. Okay, where the phosphorylations occur with the help of transcription factor H. What would be the happens when the phosphorylations occur? Carboxy terminal domains contain heptade repeated sequences of the amino acids, that is called tyrosine, serine, threonine, serine, proline, and threonine, serine, proline, and serine. These are the heptade sequences. Okay, these are repeated heptade sequences is found in the carboxy terminal domain, that is tyrosine, serine, proline, threonine, serine, proline, and serine. Okay, where the phosphorylation occur, when the phosphorylations occur, then RNA polymerase second leave the transcription factors. Okay, RNA polymerase, that is different transcription factors is associated with their transcription factors. Then after phosphorylation it leaves and move to synthesizing transcribing mRNA. After phosphorylation, point out this is the carboxy terminal domain found in the large subunit of RNA polymerase seconds. After phosphorylations, leave the transcription factors and move to the upstream sequences and transcribe mRNA. Okay. So the eukaryotic transcription, how do it takes place? You know that so the RNA polymerase seconds and the transcription associated factor, transcription factor D, points out that transcription associated factors, tata binding proteins, as like transcription factor second D, these are the general transcription factors. General transcription factors, and another is regulatory transcription factors, that is either binds to the enhancers. This is called and the general transcription factors is the most important to transcribing mRNA. Okay, and today you again you will see that's how the termination of the transcription factors would be occurs. Okay, terminations, and also I have described that how mediators act as to improve the transcription factors. So again you will see that's how termination would be takes place in the eukaryotes. Okay, that is, you have know there are three types of RNA polymerase. RNA polymerase second, first, and third in the eukaryotes. First transcribe rRNA, third transcribe tRNA, U6 snRNA, that is involved in splicing. Okay, and also that's transcribed mRNA, line, and so on. Also it's transcribed sign. Okay, then all these polymerases need two terminations because all transcribed, so require terminations. This is row dependent termination. Dependent terminations of RNA polymerase first. Okay, that is row dependent, similar to the bacteria. In the next lecture I would be described about the bacterial transcriptions. Okay, so this is the row dependent RNA polymerase first, and third involve row independent process, that is row independent process. Okay, why? Because there is the poly U stretch. Poly U stretch is found where the transcription would be occurs. That is the most complex process of the transcription in eukaryotes, and RNA polymerase second also most complex process to the terminations. Okay, because here polyadenylations in the 3 sides of the mRNA, polyadenylations occur, and then breakage of the sites would be occur and the termination would be occur. That is the most complex process. So there is the polyadenylation process would be occur, and the cutting of the site of the polyadenylations and termination would be occur. So this was the terminations process in eukaryotes. And so what is the inhibitors of the eukaryotic transcription factors or eukaryotic transcriptions? That is rifamycin and actinomycin, both inhibit the eukaryotic transcriptions. Okay, rifamycin binds to the beta subunit of RNA polymerase seconds, and nearly 2 to 3 nucleotides where synthesis would occur due to the binding of beta subunits by the rifamycin. That is also rifampicin, called rifampicin, binds the beta subunit of RNA polymerase second and inhibit the transcript, nascent transcript. What is the nascent transcript? Nascent means newly synthesized transcript. So it inhibits the newly synthesized transcript, that is near about 2 to 3 nucleotides become synthesized and then inhibited by the rifamycins. Okay, so this is the eukaryotic inhibitors. Okay, then actinomycin D, this is the most important. Actinomycin D is hydrophobic, okay, inhibitors, and then it binds to the DNA sequences, not RNA sequences. But where binds actinomycin D? This is actinomycin D. That is the minor group sequences, minor group of the GC rich sequences, where GC, it binds adjacent to the GC sequences, actinomycin D. Okay, it enters easily within these sequences and it's become hydrophobic, and then there is the 2 rings is found. There is 2 rings is found in the actinomycin D, which easily enters and make problems to the RNA polymerase seconds for the movings, because here if binds actinomycin D, distort DNA sequences, and then RNA polymerase second cannot move due to the distort of the DNA sequences. Okay, due to the distort of the DNA sequences, RNA polymerase cannot move, and further the synthesis or transcription would be not takes place by the RNA polymerase seconds, because RNA polymerase seconds need to move for the transcriptions. Okay, so why these are the inhibitors in eukaryotic transcriptions of the eukaryotic transcriptions. Okay, and so also this was the row dependent, row independent, and different pathway of termination of eukaryotes. Okay, and so also the regulatory sequences of eukaryotes, that like enhancer, but enhancers is not found in the prokaryotes. Okay, so hope this lecture would be beneficial for your career and for your futures. If you like this lecture, then please click on subscribe button and also share for your friends. Thank you. Now, what is the role of activator and co-activator in eukaryotic transcriptions? Okay, then there is the 2 types of the activator and co-activators, that is chromatin modeling complexes and chromatin modification complexes and chromatin remodeling complexes. Chromatin modification complexes and chromatin remodeling complexes. Chromatin modifications complexes are cyclic AMP response element binding protein that is similar to the HAT, histone acetyltransferase, and this is the first, and second is also self histone acetyltransferase. What is the role of histone acetyltransferase? These are the histones, okay, where arginine, lysine residue is found, that is positively charged. Okay, DNA is wrapped around histone due to the phosphate group's negative charge and lysine group's positive charge. Then positive negatives interact and the genes becomes suppressed, that is not exposed to the binding of RNA polymerase second, because there is not any exposing promoters for the binding of RNA polymerase seconds, so the transcription would be repressed. But histone acetyltransferase transfer the OCH3 groups that become negatively charged in the lysine, either arginine residue. Then the negative and negative repel to each others, exposed of the promoter region of the DNA. If they expose of the DNA for binding of RNA polymerase seconds, then the transcription would be starts. So this is the chromatin modification complexes, that is cyclic AMP response element binding proteins and HAT. And chromatin remodification complexes, these are the SWI, first one, and SNF. These are the complexes. ISV, another complexes, and another SWI second, SNF second factors complexes that is involved in chromatin remodification complexes. The most common examples of the eukaryotic activators is the galactose 4, that's galactose 1 is an encode an enzyme that convert galactose into the glucose, that is galactose 1 converts galactose into the glucose. This encode an enzyme, but the galactose 4 act as activates the DNA. So in the yeast, where upstream activating sequence element is found, where galactose 4 binds and shares the galactose 1 genes binding sites. These activates the galactose 1 genes, and the transcription of the galactose 1s would be occur, enzyme would be formed and converts galactose into glucose. In the presence of galactose 4, why? Galactose 4 binds and help to activate the transcription factor second B. Okay, galactose 4 help to bind transcription factor second B and then activate the transcriptions of galactose 1 in several folds in comparison to the normal condition without galactose 4 enhancer. Okay, without galactose 4 activators, there is reduce of the transcription, but in the presence of galactose 4, several folds increases because it help to bind transcription factor second B to the promoter region by the galactose 1 RNA polymerase, that is the RNA polymerase seconds binds to the here and activate and transcribe the galactose 1 gene. Okay, so this was the examples. And enhancer is found from the long distance in comparison to that is several thousand base pairs, maybe 1000 base pairs, 800 base pairs, but these are upstream activating sequence element. Galactose 4 found 275 base pairs long distance from the promoters, that is 275 base pairs long distance. Okay, so this activate the transcription by this way. Okay, so this was the co-activator activators and the transcription factors and RNA polymerase sequence, that is transcribe the eukaryotic transcription factors. Okay, so in the next lecture I would be described about the eukaryotic prokaryotic transcription factors and what is the process of prokaryotic transcription and which RNA polymerase is involved and how regulate the transcriptions. Okay, so I hope this lecture would be beneficial for you and for your futures. If you like this lecture, then please click on subscribe button and also share for your friends. Thank you.